Hey everybody, we are going to start looking at the history of life on Earth today. So we've been talking a lot about evolution and how life has changed over time. Now we need to stop and take a minute and talk about where life started. So the big question is, how have events in Earth's history contributed to life as we know it? And so, how did life get started? Well, we have one idea, and we call it chemical evolution, and it's the evolution of life by abiogenesis. So, abio meaning without life. So, life started um, essentially with the formation of monomers. So, it was a chemical start. So, we had chemicals come together to form monomers, monomers come together to form polymers or the macromolecules. Then we've got protobiotin formation. And then you're finally the origin of self-replicating molecules, which leads to heredity. So we know a little bit about what Earth was like millions and billions of years ago. We know that there was a reducing atmosphere, and there are very simple molecules like water, methane, hydrogen, ammonia. Now, in order to form complex molecules, you need energy sources, so UV radiation, radioactivity, heat, and lightning. We have a hypothesis on the steps of chemical evolution from primitive Earth conditions from the 1920s, and then Miller and Urey in 1953 tested this hypothesis. And so the experiment was to duplicate primitive Earth conditions within the lab. And the results showed that organic monomers, including amino acids, could be built, and so suggested that under the primitive conditions found on Earth, monomers could be formed. Now, other investigators' results showed that all 20 amino acids could be found, glucose, ribose, deoxyribose, glycerol, fatty acids, nucleotides, ATP. So essentially, monomers for all of the macromolecules have been found. Now, when those monomers need to come together to make polymers, there was a problem. Monomers are going to be very dilute in concentration, and there are no enzymes to help with the bond formation. So now we need to have an answer to that question. And so we've got clay, we've got iron pyrite. These are both materials that could have served as substrate to position monomers closer together, and then metals that can act as catalysts to help those monomers come together to build those larger polymers. Or another possibility would be that those monomers contained within water, waves could have placed the monomers on beaches, and then as the water evaporated, the sun would have concentrated the solutions, and so the monomers would be in higher concentration then. Next, we've got protobiotins, and so these are aggregates of abiotically produced molecules, but they do exhibit some properties of life. So abiotic meaning they are not alive, but they do show some properties of life. So things like osmosis, fission, electrical charge are present. So protobiotins do have a membrane-like property, and so they are actually very similar to primitive cells. And so the idea is that this could have been the start for natural selection that then led to cells. So another question is, where did the energy come from to run these early cells? And we have some answers. So we've got ATP because that was one of the monomers that was formed, reduction of sulfur compounds, fermentation, hydrogen gradients. Those are all possible energy sources, but keep in mind that photosynthesis and respiration developed much later in time. All right, so now we need to get these self-replicating molecules that lead to heredity. So right now we know that DNA can be, copied into more DNA, or it can be transcribed into RNA and then translated into protein. This is a very complex process though, and actually too complex for early life. And so now we need to think, are there other forms of genetic information that could have been used? And so this leads us to the RNA hypothesis, and this says that RNA could have been the early genetic information. There's good support for this because RNA polymerizes easily, it can replicate itself, and actually RNA can catalyze reactions, including protein synthesis. So here we kind of show the RNA monomers. You can link these together to form short RNA polymers. You can have them assemble into complementary RNA, and then that complementary RNA would then serve as a template, and so now you've made a copy of that original RNA gene. We also have ribozymes, and these are RNA catalysts that are found in modern cells, and so are these a possible relic from early evolution? They could be. So molecular cooperation deals with the interaction between RNA and the proteins it made. And so proteins that are formed by RNA may actually serve as RNA replication enzymes. So the RNA is replicated, it makes these proteins. These proteins then help the RNA by becoming replication enzymes, so they speed up that process. So molecular cooperation does work better inside of a membrane, and so RNA benefits actually from the proteins that it is making. So what we're looking at here is sort of a molecular free-for-all, as it's labeled. So this is a prebiotic soup of organic molecules. So all these things are just sort of floating around in solution. So you've got the RNA, a polypeptide that is made, and now this polypeptide can help make more RNA. That is possible within this prebiotic soup, but exclusive cooperation is going to be much more easily attained when you have a membrane surrounding this and keeping, the RNA and the polypeptides close to each other. So selection would have favored RNA and protein complexes inside membranes, as they were most likely to survive and reproduce, because all the materials that you need to survive and reproduce are going to be found closely to each other, or closely related to each other. So then DNA would have developed later on as genetic information. RNA works in this hypothesis, but DNA is actually going to be more stable than RNA. Okay, so that is our first view. An alternate view is that life developed in volcanic vents. So volcanic vents could easily supply the energy and chemical precursors for chemical evolution. And what we know is that most primitive life forms today are the prokaryotes found in or near these volcanic vents. We have a new idea that life started in cold environments, and that the interface between liquid and solid actually allows for the concentration of materials and formation of polymers. And because it's colder, molecules will last longer as well. So now we want to talk a little bit more about modern Earth. And so we have now an oxidizing atmosphere. We obviously have a lot of life present. And so this actually prevents the new abiotic formation of life. So a quick hypothesis is life is a natural outcome of chemical evolution. So then, is life possible on many planets then within the universe? So we're not going to talk a ton about that. Just something to think about. If life here started from chemicals, could life start in other planets from chemicals then as well? So now we want to talk about fossils, and we have already talked about fossils quite a bit, but again, these are preserved remnants or impressions of a past organism. We are going to look at 4 different types of fossils, so mineralized, fossils with organic matter, trace fossils, and amber fossils. So mineralized fossils are found in sedimentary rock, and what happens is the minerals replace the cell contents. So this is going to be really common with bone, teeth, shells, things like that. Organic matter fossils are actually going to retain the original organic matter. So like if you had plant leaves that were trapped inside of shale. And what's interesting with this is because you have that organic matter, you can sometimes actually extract DNA from these fossils. Then trace fossils are going to be footprints or other impressions, and so you don't actually have any organic matter present with trace fossils, but you can see here you've got like impressions of leaves right here, so those are considered fossils. And then you've also got amber. And so if you guys ever watched Jurassic Park, this might seem familiar to you, but you have essentially a fossil of tree resin that can preserve a whole specimen inside. This is usually going to be small insects. And in the movie Jurassic Park, it was amber fossils of, I believe, mosquitoes that they were using. So now, fossils give us a really good look into the past, but there are limitations to using fossils to kind of understand what life on Earth used to look like. First, fossilization is very rare. Fossils can be very hard to find. You're often finding, fragments of fossils, and you then have to date the fossils. So if you can find something that was fossilized and you can find enough of it to know what you're looking at, then you still have to be able to date it to know when that came from. So there's 2 general ways that you can date. Relative dating is by looking at a fossil's position in the strata relative to index fossils. And then you also have absolute dating, and this is when you can approximate the age on a scale of absolute time. So those absolute methods are radioactive dating and then using isomer ratios. So radioactive dating is an estimated or an estimation from half-life of products within the fossil. So you could look at things like carbon-14 or potassium-40. Different isotopes are going to have a different half-life, and so you can measure how much of the isotope is in the fossil to get an idea of how old it is. You can also use isomer ratios. And so you're looking at the ratio of L and D amino acid isomers. So L isomers are used by living things, and then the D isomers are those not used by living things. And so at death, the L form is going to convert to the D form. And so you can actually calculate age by looking at the ratio of L form to D form isomers, as long as you're taking into account the temperature as well. So now what all do fossils tell us? They give us the geographical distribution of organisms and how it's changed over time. And this is really important because Earth has changed over time. All right, and so key events that we want to kind of talk about are the origin of life in single-celled organisms. We want to look at photosynthesis and the oxygen revolution, the first eukaryotes, the origin of multicellularity, the Cambrian explosion, colonization of land, and mass extinctions and radiation. So this is a lot that we're talking about. So now, origin of life dates back to about 3.5 billion years ago. We have stromatolites as fossil evidence, so we've got prokaryotic cells as well, and they are reproducing asexually only. With the photosynthesis and oxygen revolution, we had the early atmosphere that was a reducing atmosphere. Oxygen was later produced by photosynthesis, and oxygen caused iron precipitation or rusting, and oxygen levels today are up by close to 30%. And so the result of this is that you favored aerobic respiration, and you actually lose a lot of anaerobic life forms as this happens. Now, the first eukaryotes appeared about 2.1 billion years ago. We have this idea called serial endosymbiosis as a model to suggest how. And this model says that you have folding of cell membranes to create nuclear and endomembrane systems. You can also have the engulfing of aerobic heterotrophic prokaryotes, and the engulfing of photosynthetic prokaryotes. So essentially you've got kind of folding of cell membranes to create internal membranes, and then you also have cells that could engulf other cells and kind of have them work inside of this newer, larger cell. So evidence for this are circular DNA without nucleosomes in mitochondria and chloroplasts. So mitochondria and chloroplasts have their own DNA. They also have their own small ribosomes. Now, multicellularity started about 1.2 billion years ago, and this is the time of snowball Earth, when the Earth looked like a snowball. And then diversification started only about 565 million years ago, when Earth then thawed out. Around 535 to 525 million years ago, we had the Cambrian explosion. So this was an explosion of lots of life forms, especially large predators. You had many prey capturing adaptations and defense mechanisms that we can now see in fossils around that time. Colonization of land. So when did life move from water to land? Plants, this happened about 420 million years ago. Animals, this was about 365 million years ago. Now, we also have a lot of mass extinctions and adaptive radiations. So fossils indicate that life on Earth has shifted pretty regularly, and that actually most life that was at one point alive on Earth has gone extinct. And so we've got also severe climate change that have happened. And we've got a few reasons for this. One is continental drift. So the Earth's crust plates have moved over time, and drift is actually correlated with events of mass extinctions and adaptive radiation. This is just kind of showing you how the continents have shifted over time, how the plates have shifted over time. And again, we've got 245 million years ago up until today, sort of how continents have changed. So the result of the plate movement, we've got geographical isolation, we've got new environments that are formed, old environments that are lost, and keep in mind that as environments change, so do life. Now, we also have mass extinctions to look at. This is going to be the sudden loss of lots of species over geological time, and it could be caused by asteroids or other disasters. So we're going to skip over some of these mass extinctions. And what I want you guys to know are the results. You end up with climate change, you have areas that are open for adaptive radiation, and now you've got new species that are formed very quickly. So in your own words, tell me why you think mass extinctions are important for evolution. All right, I know that was fast, guys, but you can pause me and rewatch. All right, thanks.